I. The importance of breeding cannabis. A. Definition and goals of cannabis breeding. Cannabis breeding refers to the deliberate process of crossing different strains of the cannabis plant to create offspring with desired traits. The primary goals of breeding cannabis are to develop new and unique varieties that exhibit improved characteristics, such as potency, yield, flavor, aroma, and resistance to pests and diseases. Through selective breeding, breeders aim to enhance the overall quality and marketability of cannabis strains. B. Significance in producing new and unique strains to meet consumer demands. The cannabis market is constantly evolving, driven by changing consumer preferences and demands. Breeding plays a crucial role in meeting these demands by creating new and unique strains that cater to specific preferences and needs. Consumers may seek cannabis varieties with higher potency, distinct flavors and aromas, or specific medicinal properties. Breeders respond to these demands by developing strains that possess the desired attributes, helping to meet the diverse requirements of consumers. Furthermore, breeding allows for the development of strains that are adapted to different growing environments, climates, and cultivation methods. This flexibility enables cultivators to optimize their yield and quality, whether they operate indoors, in greenhouses, or in outdoor settings. By breeding cannabis, growers gain access to a wide range of varieties that are tailored to specific cultivation conditions, enhancing their ability to produce high-quality cannabis efficiently. C. Considerations for successful cannabis breeding. Successful cannabis breeding requires careful consideration of several factors. These include 1. Genetics. Understanding the genetic makeup of different cannabis strains is crucial for effective breeding. Breeders need to be familiar with the genetic traits they wish to combine and the inheritance patterns associated with those traits. This knowledge helps in selecting appropriate parental lines and predicting the traits that offspring are likely to inherit. 2. Phenotype and genotype evaluation. Breeders must have the ability to evaluate both the observable traits, phenotype, and the genetic composition, genotype, of cannabis plants. This evaluation process involves meticulous observation of plant characteristics, such as growth patterns, leaf structure, flower morphology, terpene profiles, and cannabinoid content. It may also involve genetic testing and analysis to identify specific markers associated with desired traits. 3. Selective breeding. Breeders must carefully select the parent plants to ensure they possess the desired traits and genetic characteristics. This selection process involves evaluating multiple individuals and choosing those that exhibit the desired attributes most prominently. It is essential to consider the overall genetic diversity and compatibility of the parental lines to increase the chances of producing desirable offspring. 4. Progeny testing and selection. After the initial cross-pollination, breeders need to evaluate and select the progeny that display the desired traits. This process often involves growing a large number of offspring and subjecting them to rigorous testing and evaluation. The selected plants can then be used for further breeding or brought to market as new varieties. 5. Patience and persistence. Breeding cannabis is a time-consuming process that requires patience and persistence. It can take several generations of breeding and selection to stabilize and refine desirable traits. Breeders must be committed to the long-term process of improving their strains and not be deterred by setbacks or challenges along the way. By considering these factors and employing effective breeding techniques, cannabis breeders can develop unique and superior strains that satisfy consumer demands, enhance cultivation practices, and contribute to the overall advancement of the cannabis industry. 2. Breeding strategies for genotype development. A. Highly homozygous genotypes. Highly homozygous genotypes are achieved through specific breeding strategies that promote genetic uniformity and stability. These strategies include 1. Full sibling crosses and the production of inbred lines. By crossing two genetically similar individuals that are full siblings, breeders can increase the likelihood of offspring inheriting identical alleles. This process, known as full sibling mating, is followed by multiple generations of self-pollination or backcrossing to the parental line. This approach promotes the accumulation of homozygous alleles, resulting in inbred lines that exhibit genetic uniformity. 2. Repeated selfing to establish genetically uniform and stable lines. Selfing, or self-pollination, involves fertilizing a plant's own flowers, leading to offspring that are genetically similar to the parent. Through repeated selfing over multiple generations, breeders can select for and stabilize desired traits while minimizing genetic variation. This process ultimately leads to the development of genetically uniform and stable lines, known as pure lines or inbred lines. Advantages and characteristics of homozygous genotypes. Genetic uniformity. Homozygous genotypes exhibit a high degree of genetic uniformity, meaning that the majority of their genetic material is identical. This uniformity allows breeders to predict the traits and performance of offspring more accurately, leading to consistent and reproducible results. Trait stability. Homozygous genotypes tend to display more stable traits across different environments and generations. The expression of traits is less influenced by genetic interactions, resulting in greater predictability and reliability. Preservation of desirable traits. Highly homozygous genotypes help preserve and fix desirable traits. Once a trait is established in a homozygous state, it is less likely to be lost or diluted during subsequent generations of breeding. B. Exploiting heterosis through highly heterozygous hybrids. Heterosis, also known as hybrid vigor, refers to the phenomenon where the offspring of genetically diverse parental lines exhibit superior traits compared to their parents. Breeding strategies to exploit heterosis and produce highly heterozygous hybrids include 1. Two-way crossing between genetically divergent parental lines. Breeders cross two genetically diverse parental lines, often from separate populations or varieties, to generate the first filial, F1, hybrid generation. These hybrids possess a combination of traits from both parent lines and often display increased vigor, biomass, development, and fertility. 2. The concept and benefits of heterosis. Heterosis arises from the complementary interactions of different alleles in the hybrid offspring. It results in the expression of enhanced traits, such as increased growth rate, disease resistance, yield, and overall fitness. Heterozygous hybrids often outperform their parents and exhibit superior performance in various environments. 3. Generation of F1 hybrids with enhanced biomass, development, and fertility. The F1 hybrids produced through two-way crossing typically exhibit improved traits compared to their parents, including enhanced biomass production, faster growth, improved reproductive capacity, and increased vigor. These hybrids can be utilized to maximize productivity and quality in commercial cannabis cultivation. Breeding highly heterozygous hybrids allows breeders to exploit the benefits of heterosis and combine desirable traits from different genetic backgrounds. These hybrids often exhibit superior performance and can be valuable in commercial cannabis production for their increased vigor, yield potential, and adaptability to diverse growing conditions. 3. Selection of parental lines and progeny testing. A. Identifying and choosing genetically divergent and uniform parental lines. The selection of appropriate parental lines is a crucial step in cannabis breeding. To achieve desired outcomes, breeders aim to identify and choose genetically divergent and uniform parental lines. This process involves 1. Genetic divergence. Breeders seek parental lines that possess distinct genetic backgrounds. Genetic divergence increases the likelihood of generating offspring with a wide range of genetic variation, providing opportunities for novel combinations of traits. 2. Uniformity. While genetic divergence is important, breeders also consider the need for uniformity within each parental line. Uniform parental lines have consistent genetic makeup, which facilitates the identification and selection of desired traits. Uniformity helps breeders focus on specific traits during the breeding process and minimizes the influence of unwanted genetic variations. B. Conducting progeny tests to estimate specific combining ability (SCA). Progeny testing involves evaluating the performance of offspring resulting from controlled crosses between selected parental lines. Progeny tests provide valuable information about the genetic potential of individual plants and their ability to pass on desirable traits. Specific combining ability (SCA) is a measure used in progeny testing to estimate the additive and non-additive genetic effects. 1. Additive genetic effects. Additive genetic effects refer to the contribution of individual genes that act independently to influence a trait. By analyzing progeny test results, breeders can estimate the additive effects of parental lines and identify those with superior additive genetic value for specific traits. 2. Non-additive genetic effects. Non-additive genetic effects arise from gene interactions that occur when different alleles from each parent interact to influence a trait. Progeny testing allows breeders to assess the non-additive effects, including dominance and epistasis, and understand the potential for combining parental lines with complementary genetic effects. Progeny testing provides valuable insights into the performance and potential of offspring, helping breeders make informed decisions about the selection of superior individuals for further breeding. C. Implementing diallel design for comprehensive evaluations. Diallel design is a comprehensive breeding approach that involves crossing several parental lines in all possible combinations. This design allows for the evaluation of both general combining ability, GCA, and SCA among parental lines. 1. General combining ability, GCA. GCA represents the average performance of a parental line when crossed with different lines. By evaluating GCA, breeders can identify parental lines that consistently contribute superior traits to their offspring across various crosses. Parental lines with high GCA are valuable for establishing breeding populations or developing inbred lines with desirable traits. 2. Diallel crosses and SCA. In a diallel design, each parental line is crossed with every other parental line, resulting in a set of hybrid combinations. Evaluating these hybrids allows breeders to estimate SCA, which indicates the specific combination of parental lines that leads to superior performance in certain traits. SCA helps identify complementary genetic interactions and potential hybrid combinations for further breeding and commercial cultivation. By implementing diallel designs and evaluating GCA and SCA, breeders gain a comprehensive understanding of the potential of different parental lines and their interactions, facilitating the selection of optimal combinations for subsequent breeding programs and the development of improved cannabis varieties. 4. Breeding techniques and practices. A. Sex reversal for controlled pollination. Sex reversal techniques play a vital role in cannabis breeding by enabling breeders to achieve controlled pollination and manipulate the sex of plants. This allows for targeted breeding efforts and the production of desired offspring. Two commonly used sex reversal techniques are 1. Ethylene-induced feminization of male plants. Ethylene, a plant hormone, can be applied to induce female flower development in genetically male plants. By treating male plants with ethylene at specific stages of development, breeders can trigger the expression of female traits and obtain feminized plants. Ethylene-induced feminization is widely used in cannabis breeding to ensure a consistent supply of female plants for seed production. 2. Silver thiosulfate for masculinization of female plants. Silver thiosulfate, STS, is a compound that can be used to induce male flower development in genetically female plants. By applying STS to female plants during their early flowering stage, breeders can stimulate the expression of male traits and obtain masculinized plants. This technique allows for controlled pollination with specific male plants, facilitating the production of seeds for targeted breeding purposes. B. Selection and propagation of offspring. After controlled pollination, breeders must select and propagate offspring that possess the desired traits and genetic characteristics. This involves careful evaluation and propagation techniques to maintain the desired genotypes. Two common practices in this regard are 1. Self-pollination and the production of completely female progeny. Self-pollination, also known as selfing, involves pollinating a female plant with its own pollen. This results in the production of progeny that are genetically identical to the parent, carrying a high degree of homozygosity. Selfing can be advantageous for fixing desired traits and establishing genetically uniform lines. In cannabis breeding, selfing is often employed to create inbred lines with consistent traits and stable genetics. 2. Selecting offspring based on desired traits. Breeders carefully evaluate the progeny resulting from controlled crosses and select individuals with the desired traits. This selection process involves assessing various characteristics such as growth patterns, cannabinoid profiles, aroma, yield, and disease resistance. By choosing offspring that exhibit the desired traits, breeders can continue the breeding process with a focused and targeted approach, working towards the development of improved cannabis varieties. 3. Multiplication through cuttings to maintain desired genotypes. Once desirable offspring have been identified, breeders can use vegetative propagation techniques, such as taking cuttings or clones, to maintain the genetic integrity of those individuals. This method ensures that the selected genotypes are preserved and propagated without genetic variation introduced through sexual reproduction. Cloning also allows for the rapid multiplication of plants with the exact genetic makeup of the selected individuals, providing a means for consistent production of plants with desired traits. By employing sex reversal techniques, selecting offspring based on desired traits, and utilizing propagation methods like self-pollination and cloning, cannabis breeders can effectively manipulate the genetic composition of their breeding populations and propagate plants with desired traits, ultimately advancing the development of new and improved cannabis varieties. V. Field testing and evaluation. A. Field trials to assess performance and desired traits. Field testing is a crucial step in the cannabis breeding process, as it allows breeders to evaluate the performance and expression of desired traits in real-world growing conditions. Field trials involve planting and growing the selected progeny in outdoor or greenhouse environments to assess their adaptability, productivity, and overall performance. During field trials, breeders carefully design and set up experimental plots to ensure accurate and reliable data collection. They consider factors such as soil composition, climate, irrigation, and pest control measures to create conditions that mimic the target growing environment. This enables breeders to observe how the selected progeny respond to various environmental factors and identify those that exhibit superior performance and the desired traits. B. Monitoring growth, yield, disease resistance, and other phenotypic characteristics. In field trials, breeders closely monitor the growth, development, and phenotypic characteristics of the selected progeny. This involves regular observations, measurements, and data recording throughout the growing season. Key aspects that breeders assess include 1. Growth patterns. Breeders observe and measure plant height, branching structure, leaf size, and overall vigor to determine the growth potential of the selected progeny. 2. Yield. Breeders evaluate the productivity of the plants by measuring parameters such as flower or seed weight, total biomass, and cannabinoid content. Yield is an essential consideration for commercial cultivators, and breeders aim to develop strains that offer improved and consistent yields. 3. Disease resistance. The susceptibility or resistance of the selected progeny to common pests, pathogens, and diseases is assessed. Breeders look for plants that exhibit resistance or tolerance to prevalent pests and diseases, reducing the need for chemical interventions and enhancing overall crop health. 4. Flowering time. The duration between planting and the onset of flowering is carefully monitored. Breeders aim to develop strains that have a consistent and predictable flowering time, as this information is critical for growers in planning harvest schedules. C. Refining and optimizing strains based on field test results. Field test results provide valuable insights for breeders to refine and optimize their strains. By analyzing the data collected during field trials, breeders can identify superior performing individuals, make informed selection decisions, and prioritize breeding objectives. Breeders use the information obtained from field tests to guide the next generation of crosses, focusing on combining desirable traits and improving upon weaknesses observed during field trials. This iterative process of selection, crossbreeding, and field testing allows breeders to continually refine and optimize their strains, bringing them closer to the desired characteristics and meeting the specific needs of cultivators and consumers. Furthermore, field test results contribute to the broader knowledge base of cannabis genetics and cultivation practices. The data generated from field trials can be shared with the scientific community, helping to advance our understanding of cannabis genetics, breeding strategies, and the interaction between genetics and the environment. In summary, field testing and evaluation play a crucial role in cannabis breeding by providing breeders with real-world data on the performance, adaptability, and expression of desired traits in their selected progeny. The information gathered from field trials guides breeders in refining and optimizing their strains, ultimately leading to the development of improved cannabis varieties that meet the demands of growers and consumers alike. 6. Commercialization and seed propagation. A. Successful testing and meeting desired trait criteria. Before a new cannabis variety can be commercialized, it must undergo rigorous testing to ensure it meets the desired trait criteria established by the breeder. This involves evaluating the progeny for traits such as potency, flavor, aroma, appearance, disease resistance, yield, and other characteristics that are important to consumers and cultivators. The selected progeny are typically subjected to multiple rounds of testing, including laboratory analysis, sensory evaluations, and cultivation trials. These tests help confirm the stability and consistency of the desired traits across different growing conditions and ensure that the new variety meets or exceeds the breeder's expectations. B. The process of commercializing new cannabis varieties. The commercialization of new cannabis varieties involves several key steps, including intellectual property protection, marketing, licensing, and distribution. Here is an overview of the typical process. 1. Intellectual property protection. Breeders may seek intellectual property protection for their new cannabis varieties through patents, trademarks, or plant variety protection certificates. This helps safeguard their intellectual property rights and allows them to have exclusivity in commercializing their varieties. 2. Marketing and branding. Successful commercialization requires effective marketing and branding strategies. Breeders develop unique brand identities for their varieties, including catchy names, logos, and packaging designs. They also create marketing materials, such as brochures, websites, and social media campaigns, to promote their varieties to cultivators, retailers, and consumers. 3. Licensing and partnerships. Breeders may choose to license their new varieties to other companies for wider distribution and commercialization. This involves entering into licensing agreements with interested parties who have the resources and expertise to propagate, distribute, and market the varieties on a larger scale. 4. Production and quality control. To meet market demands, the breeder or licensed partners engage in seed production and cultivation of the commercialized varieties. Strict quality control measures are implemented to ensure genetic integrity, consistency, and adherence to industry regulations. C. Seed propagation and distribution to meet market demands. Seed propagation is the primary method for reproducing and distributing cannabis varieties. Once a variety is ready for commercialization, breeders or licensed partners produce and distribute seeds to meet market demands. This involves the following steps. 1. Breeder seed production. Breeders maintain a stock of breeder seeds, which are the seeds used to produce subsequent generations. These seeds are carefully preserved and stored to maintain the genetic integrity of the variety. 2. Seed multiplication. Breeders or licensed partners engage in large-scale seed multiplication to produce sufficient quantities of seeds for commercial distribution. This may involve techniques such as greenhouse cultivation, controlled pollination, and seed harvesting. 3. Packaging and distribution. The seeds are carefully packaged, labeled, and prepared for distribution. They are often sold in specialized seed banks, online platforms, or through authorized retailers. Proper documentation and labeling, including strain name, genetic information, and any applicable regulatory requirements, are provided to ensure transparency and compliance. 4. Customer support. Breeders or their licensed partners may provide customer support and guidance to cultivators who purchase their seeds. This can include cultivation advice, troubleshooting, and ongoing support to help growers achieve successful results with the commercialized varieties. By effectively propagating and distributing seeds, breeders can ensure that their newly commercialized cannabis varieties reach a wide audience of cultivators, enabling them to benefit from the unique characteristics and traits of the varieties while meeting market demands. In conclusion, commercializing new cannabis varieties involves successful testing to meet desired trait criteria, followed by a multi-step process that includes intellectual property protection, marketing, licensing, seed propagation, and distribution. By navigating these steps effectively, breeders can bring their new varieties to market, allowing cultivators and consumers to access and enjoy the unique qualities and benefits of these carefully bred cannabis strains. 7. Tips for successful cannabis breeding. A. Importance of high-quality seeds and controlled environments. 1. Starting with high-quality seeds. High-quality seeds are the foundation of successful cannabis breeding. Ensure that you acquire seeds from reputable breeders or seed banks that maintain strict quality control measures. Quality seeds exhibit good germination rates and genetic stability, which are crucial for consistent and reliable breeding outcomes. 2. Controlled environments. Breeding cannabis often requires precise control over environmental conditions. Providing a controlled environment helps maintain stable growth conditions, prevents cross-pollination between different varieties, and reduces the risk of contamination from pests or diseases. Utilize techniques such as indoor growing, greenhouses, or isolation chambers to create the optimal environment for your breeding program. B. Patience, persistence, and multiple generations of breeding. 1. Patience in the breeding process. Successful breeding takes time and patience. It involves numerous iterations and generations to stabilize desired traits and establish consistent genotypes. Understanding that breeding is a long-term commitment will help you stay focused and dedicated to achieving your breeding goals. 2. Persistence in selection. The selection process is crucial for identifying plants with desired traits. It requires careful observation, analysis, and documentation of each plant's characteristics. Be persistent in selecting the best individuals based on your breeding objectives and discard those that do not meet your criteria. Consistent selection over multiple generations is essential for refining and improving your breeding lines. 3. Multiple generations of breeding. Breeding is an iterative process that often spans several generations. Each generation presents an opportunity to further refine and stabilize the desired traits. By consistently selecting and breeding the best individuals from each generation, you increase the chances of achieving the desired outcomes. C. Importance of proper documentation and record keeping. 1. Detailed records. Maintain comprehensive records throughout your breeding program. Document information such as parental lineages, breeding methods used, selection criteria, and observed phenotypic traits. Accurate and detailed records help you track the progress of your breeding program, identify successful crosses, and make informed decisions for future breeding cycles. 2. Data analysis. Regularly review and analyze your breeding data. Look for patterns, trends, and correlations between traits and genetic lineages. This analysis can provide valuable insights into the inheritance patterns of desired traits, guiding your breeding strategy and helping you make informed selections in subsequent generations. 3. Intellectual property protection. Proper documentation and record keeping are essential for intellectual property protection. They serve as evidence of your breeding efforts and can support patent applications or plant variety protection certificates, securing your rights to the developed cannabis varieties. By prioritizing the use of high-quality seeds, creating controlled environments, maintaining patience and persistence throughout the breeding process, and diligently documenting your work, you increase your chances of achieving successful outcomes in cannabis breeding. These tips provide a solid foundation for the development of unique and desirable cannabis strains, contributing to the advancement of the industry and meeting the demands of cultivators and consumers. Conclusion. Cannabis breeding is a complex and dynamic process that plays a crucial role in meeting the diverse demands of the cannabis industry. Through careful selection, controlled pollination, and rigorous testing, breeders can develop new and unique cannabis strains with desired traits such as improved yield, disease resistance, and enhanced cannabinoid profiles. The importance of breeding cannabis cannot be overstated, as it enables cultivators to adapt to changing market trends, satisfy consumer preferences, and contribute to the overall growth and development of the industry. Breeding strategies such as the development of highly homozygous genotypes and the exploitation of heterosis through highly heterozygous hybrids provide breeders with powerful tools to create genetically stable and high-performing cannabis varieties. The selection of appropriate parental lines, conducting progeny tests, and implementing diallel designs ensure comprehensive evaluations and help breeders make informed decisions in their breeding programs. Practices such as sex reversal for controlled pollination and the selection and propagation of offspring play critical roles in maintaining desired traits and ensuring genetic stability. Field testing and evaluation allow breeders to assess the performance of their strains under real-world conditions, refining and optimizing them based on observed phenotypic characteristics. The commercialization and seed propagation of new cannabis varieties enable breeders to bring their creations to market and meet the growing demands of cultivators and consumers. To achieve success in cannabis breeding, it is important to prioritize high-quality seeds, create controlled environments, exercise patience and persistence throughout multiple generations of breeding, and maintain meticulous documentation and record-keeping. These practices ensure consistency, traceability, and intellectual property protection, supporting the long-term success and sustainability of breeding programs. As the cannabis industry continues to evolve, breeders play a vital role in shaping its future. Their contributions lead to the development of innovative and exceptional cannabis strains, expanding the range of options available to cultivators and consumers alike. By embracing the art and science of cannabis breeding and implementing effective strategies and practices, breeders can drive advancements in the industry, unlock new possibilities, and contribute to the growth and prosperity of the cannabis community as a whole.